Hello, everybody. Welcome to my presentation number 8. Today we will be talking about the overall pathway that we've been working on for the majority of the class, basically about half of the class. My name is Lauren. Let's dive on in. All right, starting off with our glucose molecules. Glucose is generated from our lactose through lactase, and then that can also generate galactose. This galactose pathway can produce glucose 6-phosphate, which will be integrated in glycolysis here. Another sugar is sucrose. Sucrose can get broken down into fructose and glucose. Fructose gets broken down, added into the glycolysis pathway here. Glucose ultimately goes through glycolysis to produce pyruvate. So pyruvate can then enter this pyruvate dehydrogenase complex. So pyruvate can ultimately yield acetyl-CoA, which is important to integrate into the citric acid cycle. Acetyl-CoA can also come from the breakdown of fatty acids right here. So either acetyl-CoA can be turned into a water-soluble ketone body, or it can be integrated into the citric acid cycle to yield other things, such as energy for the citric acid cycle. Other ways that we can produce pyruvate, actually pyruvate first off goes into gluconeogenesis here at the bottom, and it can go through that, be produced back to glucose, and be stored. The other thing we can do is yield pyruvate from alanine. To get alanine to pyruvate and to get aspartate to oxaloacetate, we need alpha ketoglutarate. So those are both common. Both common in there. To get alpha ketoglutarate, you take glutamate, goes through this process here. These three things are naturally found in our body. They are non-essential amino acids. So our body can just kind of make them whenever they are needed. So we need alpha ketoglutarate here for this process, but we also need alpha ketoglutarate within our citric acid cycle right here. And then we also have our oxaloacetate that was produced over this way that gets integrated into the citric acid cycle as well. Or oxaloacetate can be added to gluconeogenesis right here. All right, kind of moving forward. Let's see. Oh yeah, that's right. Moving backwards, sorry. So within the citric acid cycle, like I was saying, energy can be produced and stuff like that. So it yields NADH, it yields ATP, and it yields FADH2. Our NADH and our FADH2 will ultimately yield or contribute to our electron transport chain. This will ultimately produce the hydrogen proton gradient within the mitochondria. The proton gradient is a mandatory thing for the mechanism of ATP synthase. So ATP synthase, it has this gamma subunit that kind of rotates along and causes the open spots of the mechanism to change. So wherever the open spot is, ATP will be released from it, and then another ADP and a PI will go up into that open space right there and bind, and then the gamma subunit will rotate 120 degrees again, causing a different area to be open. So that kind of just is like the major thing of ATP synthase. That hydrogen ion goes in, causes that rotation, and it converts ADP plus PI to ATP. So that is a major mechanism. All right, so we're just going to kind of backtrack. Sorry, this is kind of like all over the place. This is a way that kind of made sense in my brain. So I hope you guys are following along. All right, so going now from this pathway here, glucose 6-phosphate here in glycolysis. Glucose 6-phosphate will start the pentose phosphate pathway. So this will turn into ribose 5-phosphate. That is a major precursor to DNA and RNA that is precursor to the sugar. And then it can also be turned into PRPP through PRPP synthetase. We're going to put that on pause because the rest of this pentose phosphate pathway, like the GAP and fructose 6-phosphate, will feed back into glycolysis or gluconeogenesis. So it gets added in down there. Okay. Cool. Going on, PRPP, right here, the phosphoribosal pyrophosphate. This can go all the way over to this process here, gets integrated in, and then it'll ultimately yield inosine or IMP. IMP can then turn into ATP or GTP through these pathways here. There's a lot going on. All right. Backtracking again. So gluconeogenesis here, we have a glycerol 3-phosphate that gets produced. This glycerol 3-phosphate can feed over here. And then our HMG CoA can go through this reductase, and then it'll produce mevalonate, which will ultimately produce dimethylallyl pyrophosphate, which is a major precursor for the synthesis of cholesterol. Yeah, so that's a major pathway as well. So this was my little poster board of pieces of paper stuck together. Just thought I'd add that in, just kind of like show you guys all my arrows, kind of like where my process of thoughts were coming from. So yeah, thank you for listening to my presentation.